Good morning, students. Today we'll talk about our next topic, that is backcross and test cross. So first of all, keep your books and notebooks ready, and I hope you're staying safe, taking all precautions and maintaining the social distance. Okay? Dear students, this is the only way to beat the coronavirus. So stay safe in your homes and at the same time enjoying your studies. Right? So dear students, let's start with our topic today. First of all, let's see what is a test cross. Now Mendel basically, if you remember, he's the one who has worked mostly in this field, and he crossed tall plant from an F2 generation with a dwarf plant. Right? He crossed tall plant with the dwarf plant. This dwarf plant is the parent plant. Okay? So from F2 generation, he took a tall plant and he crossed it with the dwarf parent plant. And this cross is called as test cross. Now this tall plant can have two possibilities, right? Because phenotypically it is tall. The phenotype is tall, but we don't know what exactly is the genotype. तो हम जो ये टेस्ट क्रॉस इसलिए करते हैं so that we can easily find out the genotype of that plant. So जो tall plant हमारे पास है, it could have two possibilities. One, it is either homozygous tall, that is TT, or it could be heterozygous tall, that is TT. But we don't know which one is there. हम ये पता करना चाहते हैं कि दोनों में से प्लांट की जीनोटाइप क्या है. Phenotype we can see that the plant is tall, but we don't know the genotype. We want to find out the genotype. Okay, so we want to find the genotype of this plant. And from which generation we have taken? F2 generation. Right? Now coming to a very typical test cross. Okay, so typical test cross will have organism showing a dominant phenotype. Dominant phenotype means the dominant, suppose we are talking about tall and dwarf. So what is the dominant phenotype? The tall. Tall is a dominant phenotype, right? And dwarf is a recessive phenotype. So if the parent, or you can say if the plant is showing dominant phenotype, it is to be crossed with recessive parent plant. Okay, it is to be crossed with recessive parent plant. Okay. The parent plant could have two possibilities, that is TTN, either it is homozygous or it is heterozygous. And it is to be crossed with the dwarf plant, and the dwarf plant will have a genotype small t, small t. Now these two are made to breed with each other, or they are hybridized with each other, and they will produce a progeny. They will produce their children. Now we'll analyze this progeny. Okay, we'll see what is the phenotype of the plant, of these plants. And from the phenotype, we can easily make out whether it is capital T or it is capital T small t. Okay, so I hope you are clear with the test cross. So test cross means we will cross a dominant phenotype from F2 generation with the parent, recessive parent. Okay. Okay, let's see an example now. I'm taking an example of violet flower with white flowers. Right? And I'm using, for purple flower, I'm using capital W. And for the white flower, I'm using small w. Okay. We'll use capital W for the violet flower and we'll use small w for the white flower. Now we can have two possibilities. Right? We can have two possibilities again. That is, either the plant is capital W, or the plant is capital W, small w. Is it okay? Yes. We don't know which is the genotype of the parent. We only know the phenotype, that the plant has flowers violet. The violet flowers are there. In case of white, we will have small w, right? So let's take up these separately. First we'll see capital W. First we'll go with the first probability. Okay. And then we'll see the second probability, that is capital W, small w with. So we can have two possibilities in this cross. And we'll always have these two possibilities, right? Now let's take it, the first possibility. The flowers are purple and we are assuming that it is capital W. And here the parent is the dwarf, that is capital small w. So we have two gametes possible here. One is and both of them will be same, capital W. And in case of small, here it will be small w and small w. So the result is that in F1 generation we will have a combination capital W, small w. Means all flowers will be purple, right? All flowers will be purple. Since all flowers are purple, we can infer all flowers are purple, so we can infer that the parent is homozygous dominant. The parent is homozygous dominant. Right? The parent is homozygous dominant. ऐसा हम कब कह सकते हैं? जब हमने क्रॉस किया है, पर्पल फ्लावर को हमने व्हाइट फ्लावर के साथ क्रॉस किया है, and we observe that in F1 generation all flowers are purple. Only then we can say that the flowers has homozygous genotype, homozygous dominant genotype, that is WW. This plant, okay, this plant we are talking about. Second possibility, let's go to the second possibility. That is, if the parent plant is heterozygous. We know the color is purple, but we are not sure whether it is homozygous or not. So we can have this. So purple flower is crossed with white flower. Here you will have two types of gametes, WW, small w, and small w. So we'll make a test, you can say the Punnett square for the F1 generation. We'll write the gametes here, then here we'll make. And we'll see the combination here is WW. Capital W, small w, capital W, small w, small w, small w, small w, small w. So we have two white flowers and two purple flowers. when we will have 2 white flowers, 2 purple flowers? जब हम रिजल्ट में देखते हैं कि 2 white flowers हैं, 2 purple flowers हैं हमारे पास, इसका मतलब है that the parent is heterozygous. ओके जो रेशो हमें यहाँ मिल रही है that is 1:1, यानी 2:2, 1:1, right? So in this case, the genotype of the parent, or you can say the hybrid that we have taken, is heterozygous. But if we get all flowers purple, the genotype is homozygous. The genotype is homozygous. So from test cross, we can easily find the genotype of the dominant plant, dominant phenotype plant present in F2 generation. So ये क्रॉस हम करते हैं F2 के प्लांट को ये पता करने के लिए कि उसकी जीनोटाइप क्या है. ओके क्लियर है? ओके लेट्स कम टू द नेक्स्ट. That is, okay, now see, this is a diagram which is mentioned in the NCERT, and this diagram exactly depicts the same that we have just discussed. Like, there is an F1 generation, the flower is purple, but we don't know what is the genotype of the plant, of the plant. Okay, phenotype, we know that the flower is purple, so it could be either WW or it could be W or small w. So we are going by the possibilities that we have discussed. If it is capital WW, so we will have same type of gametes. The white, we are crossing the recessive parent, small w, so we will have all the purple flowers, means the parent is homozygous dominant. In second case, if it is this one, we are crossing it with the small w, and again you will have 2:2 ratio. So 2 white flowers, 2 purple flowers. So 50% of the flowers are white, 50% of the flowers are purple. So this ratio you will get in when this F2 plant is in heterozygous condition. Okay. If there is any doubt, you can communicate. Or are you clear with the topic? Okay. Let's start with the next one. That is your back cross. Back cross is a cross when the F1 generation is crossed with one of the parents. Okay, when F1 generation is crossed, one of the parents. Now ये हाइब्रिड जो है, हाइब्रिड कौन से हाइब्रिड की हम बात कर रहे हैं? So we have taken a pure tall plant, we have crossed it with the pure recessive plant, and we'll have the F1 generation capital T small t. We're talking about this hybrid. Okay, we're talking about this hybrid. So when we are crossing this hybrid with any of the parent means the parent could be this one or this one, right? That will be your back cross. I hope you're getting some questions in the mind. Test crosses when we cross it with the recessive parent, and back crosses when we are crossing with any of them, right? There we have F2 generation. Here we have individuals from F1 generation. So we can say basically that a test cross is also one of the back cross. Test cross is also a back cross, or you can say it is a special type of back cross, right? It is a special type of back cross. Is it clear? So we are crossing. So in this, we had taken first parent is capital T, second parent is small t. We've got an F1 generation, which is capital T, small t, which is a hybrid or in heterozygous condition, and we will cross this F1 hybrid with any of the parents. So again, we can have two possibilities. Clear here? We can have again two possibilities. Now we'll check the two possibilities again. Okay, so hybrid is to be crossed with any of the parents. We have to cross this hybrid with the parent. Now you have two. You can have two parents. It could be the you can say homozygous recessive parent, or it could be the homozygous dominant parent. So आपका parent दोनों में से कोई भी parent हो सकता है. So we have two possibilities. So हमने F1 hybrid लिया है, capital T, small t, and we are crossing with the recessive. This is your possibility number 1. Okay. Now see, we'll have again two gametes from the hybrid, capital T, small t. The parent will have small t. So we will calculate by using the Punnett square. The F1 generation will be calculated. So you will have 4 possibilities: capital T, small t. Okay. So tall is too tall, too short. So you have 50% tall, 50% short. Right? This part is exactly similar to the test cross. Just now we've discussed. Right? Okay, so your first possibility resembles your test cross. One of the possibilities of the test cross. Very good. And remember, test cross is a special type of back cross. So all test crosses they are back crosses. Right? But all back crosses are not test crosses. Only this first possibility is your test cross. Right? याद रहेगा, जितने भी टेस्ट क्रॉस हैं वो सभी बैक क्रॉस होते हैं, लेकिन जो बैक क्रॉस हैं वो सारे टेस्ट क्रॉस नहीं होते हैं. ओके. Coming to the second possibility, this is your second possibility. So you have a hybrid capital T, small t, and you have the dominant parent plant, that is capital T. So you have two gametes here, capital T, small t. You will have two gametes here, capital T. Now we'll again calculate you can say we'll have the F1 generation, and we'll use Punnett square because it will help us to in the calculation. It will make our calculation easy. So we'll write one of the gametes here, and we'll write one of the gametes here. So capital T, small t, capital T. So here you see the combinations capital T, capital T. Again the same combination. Here there is capital T, small t. Here it is capital T, small t. Okay, it is your capital T, small t. Now if we see, all parents are tall. The phenotype of all the parents is tall. There is no dwarf at all. So all the plants are tall. So the probability of getting the tall plants is 100%. And the probability of getting the dwarf plants in the F1 generation is nil. So there is no dwarf plant here. Now you see, you compare both the, you can say the results. In this case, 50% are tall, 50% are short. But in this case, 100% tall plants are there. Okay. So this is a basic difference in these two. Right? And I hope you've understood the difference between the back cross and the test cross. Right? Let's go to your homework for today. First of all, you'll make notes of the topic. You will read the page numbers 74 and 75, and you will underline all the main points when you're reading it. You will solve, or you can write the answer of question number 5. In addition to this, there is a board question which has been asked. I'm telling you that board question. That is only one question that has been asked. The question says, all back crosses are test crosses. Okay. All back crosses, all test crosses are back crosses. All test crosses are back crosses. But all back crosses are not test crosses. Explain. Okay, I'm repeating the question. All test crosses are back crosses, but all back crosses are not test crosses. Explain. And this question has been asked in 3 marks. So it is one of the very important questions. Okay? Okay, everyone. So you will complete and will send your work to me. And if there is any query, we'll discuss it. So that's all for today. Goodbye, everyone.